Hi, I'm Jared. We are part of the biology class of DNHS. We've constructed a miniature biome in order to represent a full-life ecosystem but on a much smaller scale. Since we did not use the mini biome we constructed, we do not have an exact estimate of what was in the biome at the start of the 3-week period. I was responsible for recording the aquatic data chamber. We recorded the volume of the water at the start. It was 1,800 cubic centimeters. At the end of the second week, it was 1,600 cubic centimeters, and by the final week, the volume of the water was 1,400 cubic centimeters, decreasing a total of 400 centimeters. Some qualitative data we noticed that the water over the 3 weeks got more and more cloudy and opaque. Luckily for us, the fish we had lived through the 3 weeks, but the fish's movement had decreased, suggesting that the water could have been slowly killing the fish. We believe the fish was kept alive because the plants in the aquatic chamber released oxygen through photosynthesis, giving it plenty of dissolved oxygen. The conductivity increased, causing more ions to form in the water, making the water more toxic for the fish. Another reason for its slowed down movement would be that the pH dropped slightly from 7.08 to 7.03, which could have also made the water an inhabitable environment. The fish also had other microorganisms called protozoans, which it consumed to stay alive. The producer in the aquatic chamber was the plant. The plant created oxygen through the process of photosynthesis. The oxygen was taken in by the betta fish, known as Mufasa, through the process of cellular respiration. The betta fish's function was to serve as a secondary consumer. The fish consumed microscopic protozoa that fed on the aquatic plant.
Hey, I'm Mike. I was in charge of the terrestrial chamber. We had a plant, about 3 crickets, a garden spider, and worms. Some data we had was that the soil temperature decreased over the 3-week period, most likely due to the water transferring from the aquatic chamber to the terrestrial chamber. The air temperature basically stayed at an average of 29.5 degrees Celsius. The O2 levels increased because of the plant's photosynthesis process. The CO2 levels decreased due to the crickets' deaths in the terrestrial chamber. The crickets would have been the primary consumer, but they drowned in excessive water from the wicking of the rope in the aquatic chamber during the early weeks of the experiment. The garden spider would have been the top consumer, but had no visible food. So the garden spider left the chamber in search for other food.
Hey, I'm Megan, and I was in charge of the decomposition chamber. The chamber held mealworms and earthworms, in addition to decomposing vegetation. The mealworms and earthworms fed on the rotting material. Over time, black mold, a type of fungus, grew in the chamber. It seemed that the worms were also consuming the fungus. The decomposing material was rotting because bacteria was present in the air.